Thank you very much. I now have the great pleasure of welcoming and introducing Dr. Kanta Subbarao from the National Institutes of Allergy and Infectious Diseases. We've given Kanta a rather challenging assignment, which is in a very brief period to cover the field of virological research. The purpose of this set of comments we've asked is not literally to cover the field, but to selectively discuss and illustrate the ways in which virological research depends on certain types of functions that are examined and affected in the viruses and the insights from that research and their value. So I know we're all looking very much forward to the overview that you can provide. Thank you so much, Kanta, for being with us this morning.
Well, good morning, everybody. I'm a virologist in the intramural research program at the NIH, and my lab works on all three of these pathogens. We work on influenza, we had an active SARS program, and we work on MERS as well. So the topics that I was asked to cover today are the types of gain-of-function research, what impact virologic research typically has on viruses in terms of gain or loss of function, what we know and don't know about SARS, MERS, and influenza, and can gain-of-function research help fill the knowledge gaps? Where does virologic research cross the line as defined by the US government? So the types of questions that virologists ask are, why and how does a virus infect or kill mammals? Do antiviral drugs work? And how does a virus become resistant to antiviral drugs? Do current or novel vaccines provide protection, and can the virus escape? How does the virus spread within animals, and how does the virus spread from animals to humans and between humans? Could a virus cause a pandemic? And what is the likelihood of emergence or reemergence of a virus? So we have very powerful techniques to generate viruses de novo. With both these techniques exist for flu as well as for SARS and MERS. So we can start with a cDNA copy of a genome, make an RNA, transfect that into cells, and recover an infectious virus. In addition to this technology, which is referred to as reverse genetics, we've also had a number of advances in science that are very powerful and are applied in virologic research that involve deep sequencing, where we can now study viral quasi-species at different sites. We can study virus-host interactions at a very sophisticated level. And a number of human monoclonal antibodies have been generated, so we can look at the immune response in a way that we weren't able to more than 10 years ago. And we can also now modify the host genome, and an example of this would be using the CRISPR-Cas technology. So what are the applications of this gain-of-function research in virology? We study the biology of viruses, and we can identify novel viral gene products, and good examples are the PB1F2 of influenza or the PAX gene product. Identification of the receptors that these viruses use, including the human DPP4, which is the receptor for MERS. We study virus ecology. We characterize viruses from animal hosts and identify host range determinants. We look at viral pathogenesis and identify virulence determinants. In the case of influenza, we're able to study the transmissibility of influenza viruses. And it's very important to note that there are no models to study transmissibility of MERS and SARS coronaviruses. We also can look at virus-host interactions, because disease is obviously the result of an interaction between the virus and the host. We develop animal models, and in the case of pathogens with pandemic potential, it's important to have an animal model in which there's disease as well, because to get approval to study a compound or to use a compound in humans, the animal rule that has been set forth by the FDA requires presence of disease that mimics human disease. So simple virologic outcomes are not sufficient. So a good example of this is that we had to adapt the SARS coronavirus by repeated passage in mice, and these studies were done in my lab, to induce clinical signs of disease in a mouse. Antiviral drug studies. It's important to understand the mechanism of antiviral drug resistance and evaluate the fitness cost of antiviral resistance mutations. And similarly, immunoprophylaxis and immunotherapy are gaining a lot of attention because our antiviral drugs alone, say oseltamivir, is insufficient, and we clearly recognize the need for additional options as medical countermeasures. So identification of the epitopes that are targeted by antibodies, especially broadly neutralizing antibodies, are very important, and evaluation of the virulence and fitness of antibody-resistant mutants is important. So for vaccine development, the molecular characterization of antigenic variants requires gain-of-function studies. The generation of vaccine seed viruses with appropriate properties, including enhanced yield and immunogenicity while preserving antigenicity. And examples of these studies were done in preparing an H1N1 pandemic influenza vaccine. It's important to use these types of studies to elucidate the biological basis for adverse outcomes associated with vaccine candidates, and you'll hear more about this from Ralph Varick and Mark Dennison. It's important to use these technologies to determine the molecular basis of attenuation of vaccine viruses and to determine the stability of live attenuated vaccine candidates. So in a few slides, I'm going to talk to you about how influenza virologists identify viral determinants of virulence. So one of the ways we do this is if we have two viruses that have disparate outcomes. So if you have a virus A that is lethal for mice and a virus B that is non-lethal for mice, and these are supposed to be feet up and feet down, because influenza has a segmented genome, we can generate single gene reassortants. And so on this schematic here, you could see that there's a red virus and a blue virus, and because the genome is segmented, we can generate single gene reassortants and identify which gene segment is associated with the lethality in the mouse. And then you can compare those gene sequences and then determine the role of specific amino acids by introducing single mutations or combinations of mutations by site-directed mutagenesis, and then test the virulence of those mutant viruses in animal models. So this is a very traditional approach to studying viral determinants of virulence in influenza. For transmissibility, here's an example of Ron Fouchier's experiment done with the Indonesia 2005 H5N1 virus, and the virus was administered to ferrets that were infected through the respiratory tract, and then the virus was passaged 10 times in ferrets to yield a virus that was then characterized and was transmissible. So this virus had mutations in multiple gene segments with 2 new changes in the HA that were identified. And this virus was transmissible in ferrets, but it was not more virulent than the original virus. So I'll reiterate that there's no evidence of transmissibility in any animal model for SARS or MERS. So how and why do we study evolution of viruses under immune pressure or under antiviral pressure? How this is done is that we generate a panel of escape mutants that replicate in the presence of the monoclonal antibody or in the presence of antiviral drugs. So this can be done in vitro, where you can take a virus and grow it on a monolayer of cells in the presence of an antiviral or monoclonal antibody, and then after several days you can identify escape mutants. Or you could do a similar study in an experimentally infected animal, and here I've given you an example of a ferret, where you can infect the ferret, treat with the monoclonal antibody or antiviral drug, and recover virus at the end and try to identify escape mutants. We sequence their genomes, or in the case of influenza, maybe the target gene segment, and then introduce each of the substitutions individually or in combinations into a reverse genetics-derived virus, and then examine the fitness and resistance phenotypes in vitro or in vivo. Why do we do this? We do this to understand the biology of the virus. We do this to map epitopes that are targeted by the antibodies, or to develop more robust countermeasures, including antiviral drugs and vaccines. So this is a schematic that was given to me by Mark Dennison that outlines the coronavirus targets for vaccines and therapeutics. The coronavirus genome is very large. It's a 30 kb genome of +sense RNA. And on this slide are several of the important functional regions that have been defined by reverse genetics and adaptive experimental evolution. So you'll be hearing more about this from Mark and Ralph. So the second thing I was asked to focus on is what impact does virologic research typically have on the viruses being studied in terms of gain or loss of function? So I have to put in a few words about this term. The term gain of function is a term that geneticists use, but not microbiologists. It's vague and it's unsatisfactory for microbiologists. One suggested alternative that was suggested by Paul Dupré and Arturo Casadevall is ATRIP, which is an acronym for an experiment that uses one or more dual use of research of concern agents and aims to produce, or can be reasonably anticipated to alter transmission, range and resistance, infectivity and immunity, or pathogenesis. Or another suggestion is gain of virulence or intransmissibility. So what are the impact of these methods on gain of function in virology? So what are the things that we typically do, day in the life of a virologist? We passage virus in cell culture or in eggs in the case of influenza. Is there gain of function? Yes, and we usually want that. It's usually desired. Is there a loss of function? Yes, there's very often a parallel loss of function in that you may lose the ability for that virus to replicate well in another host. Now, if you use these alternative, more precise terminologies, the answer would be no. We serially passage viruses in experimental animals. This has been done for years. It's been done in order to develop animal models that have disease in addition to viral replication as an outcome. An example, as I told you, was the SARS coronavirus MA15 virus. Again, is there a gain of function? Yes, it's possible. We usually desire it. We're looking for this. And there is a concomitant loss of function in that these viruses often become less virulent or attenuated in other species. Genetic reassortment of influenza viruses. We do this on an annual basis to generate vaccine candidates. There is a gain of function in that there's better yield, and there's a loss of function in that there's a loss of virulence. As I've talked to you about antiviral and monoclonal antibody resistant mutants, they provide very valuable information on suitable antiviral strategies. There is a gain of function because we're inserting mutations in order to determine whether they confer resistance. And is there a loss of function? Yes, very often there's a reduction in fitness and virulence. Site-directed mutagenesis is another technique we use in the lab all the time. This is used to conclusively prove the molecular basis of a phenotype. Is there a gain of function? Is there a loss of function? Yes to both. So what do we know or not know about SARS, MERS, and influenza? And can gain of function research help fill the knowledge gaps? So what we do know about these viruses is we know quite a bit about the biology of the viruses. We know something about the ecology, about the reservoir in nature. We know about several of the host range determinants and virulence determinants, especially for influenza, much less so for MERS. We know a bit about the innate and adaptive immune response, although that area is constantly exploding. And we have antiviral drugs and vaccines for influenza, but not for the coronaviruses. What do we not know about influenza? What are the two really important questions in virology of influenza? And they are, why are some viruses more virulent than others? And why do some viruses spread more efficiently while others don't? These are critical for understanding both seasonal influenza as well as for assessing the pandemic potential of novel viruses. What drives the evolution of antigenic change and antiviral resistance? This is critical for development of antiviral drugs and vaccines. Are there viral targets that will not escape under immune pressure? This is absolutely critical for the development of universal influenza vaccines, which have been recognized widely as a worthy endeavor. What do we not know about SARS and MERS? Will SARS or SARS-like coronavirus reemerge from bats or other animal hosts? What are the critical host range and virulence determinants of MERS? Can we develop a candidate vaccine that is safe, immunogenic, and efficacious? Can monoclonal antibodies be used safely for prevention and treatment? And can we identify drugs that are safe and effective? These are critical because there's an ongoing outbreak of MERS, and they're critical in preparing for the possible reemergence of SARS. So you've heard about the safety and oversight measures that are in place, and this is just to remind you that we do work with these pathogens with a number of layers of regulation and oversight. So we work with MERS, SARS, and highly pathogenic avian influenza and the low pathogenicity H7N9 viruses in biosafety level 3 labs with respirators. We have medical surveillance and support measures in place in all of our labs for all of these pathogens. We have antivirals and vaccines available for flu, but not for MERS and SARS. We have select agent regulations that govern SARS and high-path avian influenza. And we consider dual-use research of concern questionnaires in developing research programs for all of these pathogens. So where does the virologic research cross the line into this gain-of-function research that is defined by the US government? So what is that line? That line currently includes all influenza viruses, SARS and MERS, includes experiments that are reasonably anticipated to increase the pathogenicity or transmissibility in any mammalian species. So there are places where the research crosses the line into this. One is the adaptation of MERS coronavirus to animal models, elucidating the molecular determinants of transmissibility by the airborne route in influenza, elucidating the biological basis for adverse outcomes associated with candidate SARS vaccines, conclusive experiments to demonstrate the biological significance of novel gene products, genetic differences between isolates from animals and or humans for newly emerged viruses that include H7N9, but also include H5N8, H5N2, H10N7, and H10N8, all of which have emerged essentially since this pause was announced, and MERS. Virulence determinants of all of these viruses as well, and understanding the molecular basis for resistance to antiviral drugs and monoclonal antibodies by studying virus evolution under immune pressure or antiviral drugs. So with the perspective of many influenza and coronavirologists are that the concerns about aerosol transmissible, highly pathogenic avian influenza viruses have expanded to include MERS and SARS. They've expanded to include research in using rodent models, and they've expanded to include all influenza viruses, including laboratory strains that are avirulent for humans, like PR8. So an open scientific discussion is needed to discuss the rationale and justification for this expansion. A risk assessment should be based on the biosafety measures and the personal protective equipment that are used, and the numbers that represent the actual experience of laboratory exposures and accidents with SARS, MERS, and highly pathogenic avian influenza viruses. So what are some of the priorities? To replace the term gain of function with microbiological terms that more precisely and appropriately define the experiments that are truly of concern. To lift the restrictions on MERS for three critical reasons. There's an urgent public health need for medical countermeasures with new cases and deaths reported every week. There's no evidence of transmission in any animal model of MERS or SARS, and the viruses are transmissible in nature. There are no laboratory-acquired infections in the US in over 10 years of work on SARS and MERS. To lift the restrictions on rodent models, because adaptation to rodents does not enhance virulence for humans. To focus only on influenza viruses that are of greatest concern, not all influenza viruses. And to lift the restrictions on characterizing escape from antibodies, antivirals, and vaccines, because these have a direct impact on public health. So I'll close by leaving you with the map that shows some of the viruses that have emerged just in 2014, including H7N9 and H10N8 in China, H10N7 in the North Sea, H5N8 in Europe, and H5N2 in British Columbia in this last few weeks, and MERS that is a continuing outbreak in Saudi Arabia. Thank you.
Would you join me here? I'd like to invite our other panelists now to please come up on the stage to join us. Thomas Briese of the Columbia University and Michael Imperiale from the University of Michigan. Welcome. Well, Kanta, you gave us an overview and more. You covered remarkably efficiently the set of experiments. You helped us think about terminology. And you also came forward with a set of conclusions of your own thinking about what the implications are for understanding both the nature and the implications of the problems as well as the research. So you've given us a wonderful basis to begin a conversation here. And Michael, I wonder if I might invite you to offer your perspective first, and then Thomas will turn to you afterward.
Thank you, Harvey. So I want to put my comments into context, and just to say that I don't have a stake in the game here. So while I'm a virologist, I've been studying viruses for over 30 years, I don't work on any of the pathogens that we're discussing here. And also, on the other hand, I have been involved in these discussions for the past decade, having been on NSABB for almost its entire existence. So what I'd like to do is touch on 4 concepts. You've heard about some of these before, and really just maybe emphasize and expand and give a slightly different viewpoint on some of these in some cases. So the first, and a number of the speakers this morning have already raised this, is that I think we've got to get rid of this concept of gain of function. You know, I think that viruses are gaining functions all the time. We know that RNA viruses have a very error-prone RNA polymerase, and so these mutations are arising all the time. And so the fact that we are deliberately imposing gain of function doesn't mean that we're necessarily doing anything that nature is not doing already. And I would mention that with respect to the terminology of the pause, you know, in terms of what can be reasonably anticipated, well, I can reasonably anticipate that anytime one grows an RNA virus in the laboratory, these mutations that have gain of function are arising all the time. And so the question is, are we going to be concerned about that because those might accidentally get out? We would shut down virology research if we took things that far. And so I am pleased to hear Sam Stanley say that the NSABB has asked the government for some guidance and clarification on what is going on there. And my final point here would be that I think that the term gain of function is being misused more than the viruses that we're worried about here today. And the problem is that, you know, we in this room all know what we mean when we say gain of function, but the problem is that when the public hears what's going on and how these discussions are going, they can't make that sort of nuanced distinction that we can make here. So I really do think we have to get to a different terminology, and I'm not even sure that a trip works. I'd rather that we just take each individual case and call it what it is, rather than trying to come up with some acronym or 2 or 3 word term that can easily be misinterpreted. The second thing I would want to point out is just my kind of view on the use of ferrets as an experimental model, because that's been debated a bit with respect to especially the H5N1 experiments that we've been talking about. And so my feeling is that they are a useful model. By and large, human influenza viruses behave the same in ferrets as they do in humans. And I would also point out that, you know, both Ron Fouchier and Yoshi Kawaoka, although they did separate experiments, they used separate genetic backgrounds for their viruses, the phenotypes of what came out really were convergent in terms of the types of things that were affected in the virus. And so to me that says that the ferret is a useful model. Now, of course, the problem with ferrets is that because these experiments are expensive to do, we can't get high numbers of animals that we infect, and so maybe that limits to some degree the statistics that we might be able to do with those experiments, but I still think that this is a powerful approach. The third point I want to make is with respect to something that Kanta also talked about, which is antibody escape mutants, or also, for example, drug resistance mutants. You know, these escape mutants are arising all the time in individuals who are infected with influenza. And so what we're doing in the laboratory is not that much different from what's occurring, again, in nature all the time. And so I would argue that, you know, there may be a different degree in selection in an in vitro experiment versus, for example, an animal model. And no animal model is perfect. That's something else I should point out. But, you know, these things are happening all the time. And so are we really creating anything in the laboratory that nature itself is not creating? And I think that that's something we need to think about. And then finally, I think the last point I'd like to make is that risk mitigation is available in virological laboratories. And so, for example, we can use strains of viruses that have been engineered such that if they get outside the laboratory, they may not be as virulent. And an example of this is some of the work that Adolfo Garcia Sastre and Ben Tenover have done at Mount Sinai School of Medicine, where they've engineered into the influenza genome the target site for a human microRNA, such that if that virus infects a human, it can now no longer replicate because the microRNA is basically going to shut down replication of that virus. So we can develop safer approaches to do these types of experiments. It just needs a little bit of imagination on the part of researchers. And then finally, we can vaccinate laboratory personnel and create sort of a ring of protection. And I know that Ron, and I'm assuming maybe Yoshi's lab also, their lab workers are vaccinated against H5N1, such if there were an incident in that lab, an accidental exposure, presumably then that would greatly lower the risk that that virus can then spread to others. And so these are my sort of viewpoints, sort of as someone who's not working on these particular viruses, but thinking about it a lot, kind of looking in. And I hope that these are some things that we can consider in the discussion.
Wonderful. Michael, thank you so much for those observations. Tom, would you like to speak from the podium? Or just get a remote? Okay, that's good.
Yes, thank you. So I'm also not really concerned with any experiments discussed here, so gain of function research. And I just wanted to, reiterate some of the points from Kanta's excellent presentation that summarized, I think, the work that is done and can be done and the questions that are open. And so I just wanted to touch on 4 areas that I think are important in the discussion. And one is, again, gain of function is a highly diverse, complex area. There are many different types of experiments concerned, and there are also many methods concerned that are used in these experiments. And these are not really particular new or special methods. It's common virologic methods that are used: propagation, adaptation, selection of organisms. And by that, it is not just also a question of virology. These methods are used in other areas of microbiology. And as such, the discussion that we are having today and tomorrow has a wider reach than just on the organisms that are mentioned here, influenza, SARS, and MERS, but also beyond virology. The next area that I want to talk about is there are, in many cases, very good reasons for doing gain of function experiments or research, and also in many cases there is no real alternative. So if you think about a complex phenotype that you want to select, that is not easily possible to construct by targeted or directed mutagenesis. So from that, if you select for those phenotypes, there is a degree of unpredictability in the experiment. So from that, it is not really clear what comes out of such an experiment. Think about drug resistance. The dilemma there is very obvious. You can ask, is it possible to generate a resistance to a particular treatment? And if so, what is then an alternative way to treat? And with that, you can take a very reactive approach, wait until it happens in nature that you find an escape mutant, then investigate it and come up with a solution. Or you can take a proactive approach, select in the laboratory, as Mike also alluded on, select for the resistance mutants, characterize it, find out the mechanism, and come up with an alternative solution. And if you just look at the phenotype and the pathogenetic overcoming of the resistance that's independent of the underlying mechanism. So in that sense, you're not dependent whether the mechanism that was selected is the only one or the correct one to come up with the resistance. That is in contrast to more complex phenotypes like transmissibility, where you have a multitude of ways to get the phenotype and where there is genetic background involved that might modulate the actual mutations that are identified. So here the question is the information that you get actually actionable information? So can you do or can you design a countermeasure based on the specific changes that have been observed? If that phenotype can also come about in different ways. So from that, third, I think gain of function research has been there for a while, for decades, and it will in some form be there in the future. The question is not whether it can be done, but how it can be done, and how can it be done in a safe manner and ethically sound. And that leads to the last point. I think the current criteria for restricting funding are somewhat counterproductive to the issue or the goal of that rule. As Mike suggested, a possible way to mitigate risk is vaccination, which is fine for flu. But for example, in case of MERS, as we heard from Kanta, there are no model systems to do vaccine testing or drug testing. So one would have to create in MERS virus that generates pathogenicity in a small animal model system that is comparable to humans, but that is currently paused under the regulation. That was it. Thank you.
Thank you very much. I wonder, Kanta, if you would have any reflection based on the comments. In particular, why do you think it's useful to have a classification that is precise enough to get to the problems that we want, inclusive enough to cover the topics that may produce concern, but stop short of what I think Michael was suggesting of really looking at each individual experiment on its own terms to make a judgment about. Could you comment on that aspect?
I'll take a stab at it.
Yeah.
So I think what you're asking is, so where would the idea of each experiment being reviewed be too cumbersome? Mm-hmm. And I think at every one of us that does experiments in laboratories, we have institutional biosafety committees, and we look at each experiment in terms of dual-use research of concern. So there are already layers of thinking about the experiment. The question is, at what point would a higher level of oversight or risk-benefit analysis be triggered? And that can be done for specific experiments that meet some sort of a line. But I think what I'm most concerned about is that this pause has swept far too many aspects of virologic research into the sort of definition. And so my plea would be to be much more precise and focus on the pathogens and the experiments that are of concern, rather than having such a broad definition that you virtually bring research in several areas to a standstill and then incrementally move it forward.
Thank you. We are now open to comment or question from members of the audience. If anyone would like to pose a question or to offer an observation, the floor is open. Okay, this is, if I may take a moment to convey a question that has come in from the viewers on the webcast. This is from an individual who works with both geneticists as well as microbiologists, and in fact works in microbial genomics, and describes the fact that gain of function as a term is really used to characterize the recent work on giving influenza the ability to transmit among mammals. But the questioner is raising the point, why is it useful to avoid the phrase gain of function? I mean, what's wrong with it is really what he's asking. So who would like to begin? Everybody's ready to jump in on this, but Thomas, why don't you start, and then we'll move it.
Yeah, I mean, as I pointed out earlier, it's a very diverse area, and many different kind of experiments and designs are covered by that, and I think that's a little difficult for the discussion. As Kanta already pointed out, it is a too wide net that some lumps up things that are particular not the concern here, I think. So we should restrict it probably on transmissibility, virulence of certain influenza types, not all influenza in general. I mean, there is a difference from risk and biologically between common circulating seasonal influenza and H5N1, for example. So from that, I think it must be much more differentiated and really targeted on the issues at stake, that you very specifically define what is mentioned. Otherwise, it becomes too difficult and too complicated.
Yeah, I think each speaker, in fact, I think has emphasized that the term is too inclusive to give you guidance on what is truly worrisome, and I think that was a key point. Let me turn now to the questioners here and or commenters, and begin over here. If you would again identify yourself, and then we'll take people on both sides.
I'm David Relman from Stanford University. Thank you for your comments. I would suggest that many agree with many of the points you have made about the importance of greater specificity in what it is we're talking about and what it is that we may be concerned about. And so I would just like to propose, for the sake of discussion, a more specific scenario in which gain of function means the deliberate creation of a combination of the following properties in the same agent: high pathogenicity, high transmissibility among mammals, and let's say as well evasion from all available countermeasures. And let's say that this is a combination that we don't think exists in nature in this particular agent. Could you address whether, A, you think this is a very particular scenario within this larger realm that might deserve both greater concern and perhaps oversight, and whether you see that there are experimental alternatives to understanding each of those individual properties without creating such a combination?
Okay, so the horror case that Dr. Relman is proposing is an experiment directed at simultaneously enhancing transmissibility, pathogenicity, and evasion of either treatment or other intervention. Would any of the panelists wish to then comment? Would this be a class of research that you would countenance, or that you think would be beyond the pale of acceptability? Kanta, you can go first.
I think this is on. So I think the experiment that you've described of generating a virus that does not exist in nature, that has all three of those properties, I think everybody would agree would be a matter of great concern, and all the regulations say it's an experiment that should not be done. And if it is done, it should be reviewed at the highest level. So we have those regulations in place. Are there alternatives to doing that by studying each separately? Absolutely. The trouble is that we don't know whether enhancing the virulence will also alter transmissibility until you study the transmissibility of the virulent agent you've generated, or the other way around, where you generate a transmissible virus and then study whether it is has also gained virulence, because the properties may or may not be conferred by the same mutations. But by intent, generating such a thing would, I think you'd find universal agreement, would be of concern.
Thomas, you want to add a word?
Yeah, I want to make maybe a comment and then go to the question. So you said transmissibility among mammals, and I think that's also already too wide of a definition. I mean, think about transmissibility among its natural host species, transmissibility to a mouse, or what we are really concerned would be transmissibility among humans. So that might also be differentiated. But if you say, let's create Frankenstein, fine. The question is there any real good reason to do so? And Kanta pointed out you might find a safer way to do it. But what I would want to touch on is, even if you would do it, what would be the risk or the problem? You have in the end an organism that is dangerous. And the real problem is if it's handled for a long time, continuously, maybe on a larger scale and multiple labs, then you run into the problem, how can you deal with the risk that it might be escaping in the community and creating disease? So the question is, how can you regulate the outcome of a gain-of-function experiment, not necessarily stop the whole research from the get-go?
In the interest of moving on, Michael, it's okay, I would like to invite the next questioner, and then we'll try to keep focused our answers as well. Please.
Thank you. My name is Türkan Gardiner, and as a statistician, I have become aware of meta-analysis, that is, using results on previous studies, designed or non-designed, in terms of interpreting a path for future research. Do you have any suggestions at this point? I personally don't, but an area or prospects for gain-of-function research in terms of evaluating previous studies that have been done on various areas?
Okay, so I think the question is there a way to learn from a set of previous studies to answer questions that would, in effect, render not as necessary an additional experiment, if I follow the
Correct.
thrust of the metaphor to meta-analysis?
Yes.
Would anyone care to comment on that? Michael, do you want to offer a view?
I'll take a stab at that. I guess theoretically there is. I think sort of off the top of my head, I'd have to think about your question more, but off the top of my head, I think that the genetics of these viruses, particularly influenza, is complicated enough, and that there are enough epistatic interactions that, while possible, I think at the end of the day you might just have to do the specific experiment to address the question. Maybe Kanta can comment.
I can just expand briefly on that. Influenza viruses are quite diverse amongst the different subtypes of influenza A viruses. So you can, if information exists on the specific subtype that you're interested in, that would be useful. But you cannot necessarily extrapolate from information based on a different subtype and a different gene constellation, because the gene segments and gene products interact with one another.
Thank you. Next question.
Hi, my name is Adolfo García Sastre. I'm working with influenza viruses. I want to have a comment. Again, thanks a lot for your presentations about the inadequacy of using gain of function as a term that may result in something that is dangerous. So I believe that what we are all concerned about is about the possibility of generating a virus that creates more danger in the lab than what is already existing in nature. And this is a gain-of-function experiment, but there are many gain-of-function experiments that do not result in viruses that are more dangerous than anything that is right now circulating in nature. And I wonder whether one could be a good way how to frame what we are worried about, and then focusing the deliberations in this particular set of experiments, and not so much in experiments that are also gain of function, but do not represent a major danger to public health.
Thank you very much. I think we can take that as a comment and even a reinforcement of some of the points that had been raised before, and I appreciate it very much. Let me come over now to this side.
So my name is Paul Dupre. I'm from Boston University. I just want to make a couple of points about the terminology that you're using. First and foremost, gain is not the only thing we should be considering. We should be considering both gain and loss, which is why any selection process involves an alteration in phenotypes. Kanta was asked about 3 separate phenotypes, all gaining function, enhancement of pathogenesis, and whatever the 3 were. And that is very, very unlikely in the sense that for a gain in function, there is also oftentimes a concomitant loss in function. The final point that I would like to make about terminology is if, and I'll use a specific example that I have used previously. I work in a virus, again, like Michael Imperiale. I don't have any skin on this. I don't work in flu. I don't work in coronaviruses. I work in other RNA viruses. And one of those RNA viruses is now in 2014 known to use 2 receptors. That's measles virus. It infects immune cells and it infects epithelial cells. Whenever the vaccine was generated, prior to knowing about those 2 receptors, that vaccine was passaged in inappropriate cell lines. A human virus passed in animal cell lines. And what we know is that virus did not just infect 2 cell types, immune cells and epithelial cells, but that measles vaccine became pantropic. It can infect every single nucleated human cell. What a gain of function of experiment. If we want to use the term gain of function, that's a tremendous gain of function. But what has that virus, that vaccine done? It has led to the near eradication globally of a virus which has a higher R0 and was killing more children than these viruses that you're discussing today in the 1960s. So again, I just reiterate the point, be very careful how you think about terminology, alterations, gains and losses in function.
Well, thank you also for that comment. Basically, again, looking for more precision in language and recognizing that functional change is not necessarily a perverse result, that it can also have beneficial results. Does anyone wish to add a comment on this? If not, we can turn to the next commenter or questioner, please.
Simon Wynne-Hobbs from the Pasteur Institute in Paris. Would the panel, we've got lots of semantic questions, but would the panel agree that essentially what we're really concerned is work on that makes viruses more dangerous for man? And when I say man, I mean man, his animals, and his plants. I've got 2 subsequent little questions to follow. Is that what we're really concerned? Get rid of the semantics, get rid of gain of function. We're worried about manipulating viruses that make them more dangerous for man. Can we agree on that?
Okay, so the question is, if you strip away the terminology and simply take consideration of experiments aimed at mutations to make an agent more dangerous to humans, is that a concern?
Is that the real group of experiments we're concerned about?
Michael.
I guess I would qualify that a little bit, Simon, and I guess the question becomes, do we want to take a proactive approach or do we want to take a reactive approach? So we could wait and see if H5N1 starts to become transmissible from human to human, or we can say, you know, this virus has been sampling the human population now for quite a time, and do we want to be proactive and ask, what should we be on the lookout for in terms of something that might give us a heads up and allow us to get ahead of the game? So that's the way I would view it.
Others wish to comment? Kanta?
I think I would also qualify that because, again, this definition of what makes a virus more dangerous, I think that you could certainly say that a virus that occurs in nature that and you've identified a potential antiviral drug, and you want to see if the same mutation in that pathogen would confer antiviral resistance, I think that would be an experiment that would be a responsible experiment to do in preparing for the use of medical countermeasures. Similarly, there's a lot of focus on developing universal influenza vaccines, most of which are directed at very specific targets in the genome, and I think it's critical to work out whether those regions of the genome are stable or not.
I agree entirely, but from what we know about microbiology, there are a very large number of microbes that can come over. They're called dead-end infections. The most well-known is rabies, 55,000 deaths per year, there's no transmission. But there are so many viruses out there that come over in small numbers. Do you think we're going to have any success in predicting which of those microbes are going to be of big concern? H10N8 with 3 cases, is not emergence. One of those cases was immune-suppressed individual. Do you think we have a track record to show that we have any success in looking into the future and predicting which ones will go big time?
Well, thank you. The second question, just to frame it directly, is do we have a track record that gives you confidence that we can do a series of studies or a study that will help define which of the extant viruses will become pandemic? Kanta, you kind of alluded to this in your presentation. Do you want to comment, and then others as well?
So we've had an N of 4 viruses that have caused pandemics in the last 100 years. And we have not been successful in predicting ahead of time which ones those would be. But we are much better positioned today in understanding what the reservoir is and what viruses exist in that reservoir. We are much better able to identify human infections. And I think as a scientific community, we can figure out, as Michael said, whether we want to wait and see when a virus emerges and respond to that, which sometimes we have to do because we haven't predicted the pandemic of 2009, or work proactively in controlled situations and develop vaccines and models against potential pandemic threats.
Thank you. Come over here to comment or question.
Thank you. Mark Lipsich from Harvard School of Public Health. I too agree that we need a refinement of the criteria. I think for the experiments that generate special concern, 18 out of about 240 NIH grants in flu and coronaviruses is restricted, but it's maybe not restricted enough. I would agree with that. The heuristic that I would suggest as one, not the perfect, but that helps me, is those experiments in which a biosafety issue is a public health issue, not an occupational health issue. In other words, in which we reasonably expect that if there were a release, it could create a public health problem rather than a problem limited to the local laboratory. An analogy is the idea of smoking. When we thought of smoking as an individual choice with an individual risk, we let it go on more or less everywhere. We now still let it go on, but because we've seen it, thanks to the work on secondhand smoke, as being a public health issue, we subject the same risk to a much greater restrictions. So there's a very small handful of experiments that fit that definition, but I think it might be a useful one.
Thank you very much for that additional comment. Let me turn.
Well, my name is Giorgio Palua. I come from Padua University, Italy, and I'm here also on behalf of the European Society for Virology. The more I attend this meeting on gain of function, the more I'm convinced that, talking about risk-benefit, the subject that are more at risk are science and virology. And, well, quite at variance with my friend and colleague Simon Way-Hobson, who just addressed the podium, I'm convinced that it's important for the sake of this meeting, and the chairman just defined that this 1.5-day meeting is not to come with a general consensus, but just with a few concepts well clarified. I think that nomenclature and semantics are not trivial argument, but they can be essential. Look, the cornice of this building, where in Greek scripts you find Sophia, Paideia, Theoria, and they have all meaning. And you say, to give a name to a subject is an essence. So nominant sunt essentia rerum, and nomina numina, like the Latin taught us to really identify the single individual God. So here we have a number of classification. If we start with dual use research of concern, according to 428 2009 regulation of the European Community, there are more than 90 pathogens as compared with 15, and does clearly apply to biosecurity, not to biosafety, because it's intended to cover the intentional misuse of knowledge and tools and trade of them that could scare and threat society and humanity. So DURC regulation can be different in Europe and the state. So DURC probably and biosecurity is something, if I have to take care of, I should really revert to my intelligence office of my country. But biosafety is something that is really impacting on the behavior that is generalized, need harmonization, and as you said, is not just a biosafety level 2, is altogether of rule and regulation and also teaching and responsibility. So we have department, institutional level, and university level, and also rule on biosafety that are in the US and also in Europe. But even risk and benefit, you know, can create a duality that is not easy to understand, because the chairman rightly said, you know, benefit or risk, they carry a different way in terminology, because psychologically risk is more powerful than benefit. And you cannot define benefit quantitatively in science like we heard. So really, the benefit of science is knowledge per se, plus that these rules are becoming too general, that I'm afraid really constraining a bit too much virology, because, you know, we cannot even more work with coronavirus if we are just adhering strictly to. So we have a better definition of risk and benefit, but also for the sake of science, I think we have in the end to conclude that for biosafety, who is going to decide? So we are proposing, and I think it's important, this discussion, also at the academy level, European, American, Australian, but in the end, we have to come with something that is not constraining science and virology, because I'm also quoting what was said today. I like very much this virological discussion. Gain of function was born with virology, culturing the virus, transmitting, and also have attention in Europe, agriculture is regulated by Brussels, but not medicine. Medicine, every country can decide. So really we have to come with quite wise decision, and also to define very well DURC, biosafety, biosecurity, risk and benefit, and GOF is certainly inappropriate as a definition.
Thank you very much for the comments both on terminology, on institutional responsibility, and on the complexity of conveying clearly and completely the issues that we are truly concerned about. Please.
Thank you. I'm Mark Dennison, Vanderbilt University. I wanted to comment as listening to these conversations. One is, I think we need to have a greater respect for our biosafety colleagues who manage our laboratories and our programs, and the issues that are associated with the management of our programs already, particularly at the fundamental level of biosafety. What an important, exceptional job they do in combination with occupational health that I think reduces risk dramatically already. Second is, I think we need to take a case-based approach, because when I hear broad terminologies like gain of function, when we begin to think of things as the research, risks of the research, I'm very concerned about an external approach that broadly sweeps those in. I would have never have considered 11 years ago that coronaviruses would be a subject for these kind of conversations. When I was sitting on the beach with my wife trying to decide our future and deciding was coronaviruses have any future in research with just causing mild human infections and no interest in those diseases. Pointing out the impossibility of our being able to predict which humble virus family will be the next potentially pandemic virus. Finally, I'd like to comment that the issue associated with predicting that which will make something dangerous in a human is quite remarkable. I've been working with these viruses over 25 years now, and am shocked with every single discovery that we make, both those that are independent results from reverse genetic experiments and those that are associated with epistatic changes. Kanta pointed out that in the passaging of the mouse-adapted SARS, that each time that experiment was performed, a different constellation of mutations was identified, and those were not in the places that you would expect. And so that I think the potential to predict that which is going to be more dangerous brings up finally the point that I'd like to be incorporated in the rest of this discussion, which is that we have to look at things in terms of where is that risk assessment going to occur. Will it be an a priori, prior to peer review, post-peer review, post-performance of experiments? And therefore, this brings me back to the final comment, which is that we have to incorporate a case-based approach. We have to understand better the biology of coronaviruses if we want to incorporate them in this process. Thank you.
Thank you very much for those comments as well. We actually have a couple of questions from our webcast participants that bear on this question of the need to do the type of research to alter viruses in the laboratory as opposed to taking fuller advantage of natural variation in viruses in the field and using those as the basis for investigation, inquiry, and discovery on each of the areas of interest. Would any of the panelists like to comment on that particular question? Why do we have to induce change when what we're looking for are changes that may affect the human population? Why not simply study the viruses that occur and evolve in nature?
So from a very practical standpoint, we certainly do usually start with what occurs in nature. And a good example are all the studies that were done to establish the virulence determinants of H5N1 viruses, and those were based on studying the original 16 isolates that we had in 1997, 1998. But on a very practical level, we don't often have access to a very wide range of human isolates that can be characterized extensively enough. And so what we typically would do is take two viruses that have sort of differing phenotypes and then explore what the basis for those phenotypes are if they are likely to be of interest to public health or biology. So usually it is a practical limitation in terms of the number of viruses that are naturally occurring that we can study. And as Michael mentioned, there's the whole phenomenon of epistasis in influenza viruses. And so in a very controlled laboratory setting, you can conclusively prove the effect of a specific mutation.
Thank you very much. There's did you want to add a word, Michael, on this point before we go on to the last question?
I guess the only thing I would add is that, if we have the appropriate public health infrastructure in place, then these experiments can be very informative.
And Thomas, please.
Yeah, I think it was also already mentioned the advantage to doing something directed in the laboratory in addition to have better control is to come to things that might not exist in nature yet. So you should look what is out there in nature, analyze it, but there are other opportunities with an experimental setup in the lab to come to variants or phenotypes that are not yet exist, and by that you are a step ahead of nature to try to find the characterization of these and measures against them.
Good. Thank you very much. One final questioner from our webcast audience wanted to raise a question, and I'm not sure any of the panelists will be in a position to answer, but I do want to mention it because it is relevant in the fuller considerations. Namely, Are you aware how many laboratory incidents in the U.S. have occurred since early work on H5N1 that have involved any exposures and dangers from either work on influenza viruses or SARS and MERS, say in the last 20 years?
Well, I guess I've been involved in studying H5N1 viruses from the first isolate from Hong Kong in 1997, and I've worked on SARS and MERS, and I'm not aware of any, but I'm obviously not the person that people report to, so I could stand corrected, but I'm not aware of any incidents that resulted in infections in laboratory workers with any of these pathogens in the United States.
Any other comment? No. Please join me in thanking Kanta and Thomas and Michael for their excellent discussion today. Thank you so much.